Chapter 17, the Special Senses. The special senses and sense organs are olfaction, which is smell, and the nose possesses the receptors for olfaction. Gustation, which is taste, that is the tongue that has the receptors. Vision, which is seeing, is the eye. And hearing and equilibrium, which both are detected by the ear. For olfaction, the sense of smell, there are two olfactory organs involved. They are located in each of the nasal cavities, and they consist of two layers of tissue. The outermost layer is the olfactory epithelium, and the inner layer is the lamina propria. The olfactory pathway is extremely sensitive and can respond to only 4 molecules of the odorant to activate the receptors. From the axons, from the olfactory receptors, those axons will pass through the cribriform plate and synapse on the olfactory bulb and then travel the olfactory nerve to the olfactory cortex of the cerebrum. This does this without passing through the thalamus, the only sensory information that does not get evaluated by the thalamus and always goes up to the cerebrum. Now, just because we have activation of those sensory neurons, the afferent fibers, it doesn't guarantee that that will make its way to the conscious awareness of smell. Convergence and inhibition occur along the pathway and affect the ability of sensation from reaching the olfactory cortex. So it doesn't have to pass through the thalamus, but it still doesn't always make its way to the cerebrum and be interpreted. Central adaptation can occur along the central nervous system, and you quickly lose awareness of smell so that you can detect new odors that are coming in. And we also can distinguish between about 2,000 to 4,000 different chemical stimuli. The olfactory epithelium contains a variety of cells. There are the olfactory receptors with their olfactory cilia. There are supporting cells, and then there are basal cells, which are stem cells. This epithelial tissue will cover the inferior surface of the cribriform plate, the superior portion of the perpendicular plate that separates the two nasal structures, and the superior nasal concha of the ethmoid bone. Below that is the lamina propria, which is areolar tissue and has blood vessels and nerves, contains olfactory glands that produce mucus, and will be right below the epithelial surface. The mucus that is produced from the olfactory glands will come out and put a layer on top of the olfactory epithelium because you need fluid for the odorants or the odor molecules to dissolve into so that the cilia can detect them and pass them along. Your olfactory receptors are highly modified neurons. Most receptors are specialized cells that then turn around and tell the neurons, but these are actually neurons. And the exposed tip that has cilia is really modified dendrites. It will project beyond the epithelial surface into that mucus layer, and as the molecules come in, the odors will bind to receptor proteins that are on the cilia, and then this will trigger the second messenger response, which will open sodium channels, and depolarization will occur, starting an action potential. Your olfactory receptors are replaced by new ones. Okay, this is the function of the basal cells, those stem cells. They'll do division and differentiation and then replace the olfactory cells as they are damaged, because it's a tough environment. If you're smelling chemicals, chlorine or ammonia, it's going to kill those dendrites and kill the olfactory receptors, so we need to make new ones. This is a very rare example of neuron replacement. The total number of olfactory receptors is going to decline with age, starting at about age 40, and you become less sensitive to the stimuli. Can't smell as well. The second special sense is gustation, the sense of taste. It is composed of gustatory receptors, which are your taste receptors located on the surface of the tongue, and they are chemoreceptors, which mean they respond to the presence of the chemical molecules. Okay. Your tongue is covered with a number of bumps called papillae, and these bumps will have a variety of jobs. Some will have taste buds for tasting food. Others will provide friction to help the tongue move the food around in the mouth so that it can form into a ball and you can swallow it. Your taste buds are going to be formed by taste receptors plus specialized epithelial cells. The adult has about 3,000 taste buds. Taste receptors are your gustatory receptor cells. Your taste buds are made up of a number of different cells. They have stem cells called basal cells. They have gustatory receptor cells that are in various states of maturity, and some other support cells. The mature gustatory receptor cells have taste hairs on their surface, and these will extend out the taste pore of the taste bud and into the surrounding fluid, the saliva. Your protein receptor molecules on the hairs are going to bind with the molecules that are dissolved in that fluid. Again, you need to have the chemicals dissolved in the fluid, or else you can't detect them. Your gustatory pathway, when you do detect something with your gustatory cell, it will be sent to the medulla oblongata, then to the thalamus, and then on to the gustatory cortex in the insula. From there, that information will also be sent to the hypothalamus and the limbic system, which are both involved with memory and emotions. This is because your sense of taste and smell are highly linked to your memory and your emotions. Now, really to taste well, you have to have a lot of involvement from smell. About 75% of your ability to taste comes from smell. You're several thousand times more sensitive to taste when the olfactory receptors are working normally than when you can't smell. So when you get a cold, you get a thick mucus layer, and your olfactory receptors can't pick up the chemicals as well. You don't taste, but you also, don't smell, but you also don't taste. Primary tastes. There are 6 different primary tastes. We have sweet. Now there's reasons for this. We need glucose for ATP production. Salty is the second taste. We need to maintain our salt-water balance. Sour and bitter are 2 other tastes. Both of those are going to be involved with the taste of toxins. Something that is bad tends to taste sour or really bitter. So for your survival, you want to be able to detect those bad things from the good things. We have 2 other tastes. Umami, which is a meaty taste, and this is going to be because you need to keep your protein content up. And the last is water receptors. Now, the threshold for stimulation varies with various receptors, but we tend to be much more sensitive to unpleasant stimuli, which makes sense, so that as soon as you get that toxin in your mouth, you spit it out, you don't eat it and then go, oh, that wasn't so good, and then you end up dying. We have more than 10,000 taste buds, but that number will decline dramatically by age 50, and then food starts to become bland. The third special sense is going to be vision. Vision is much more complicated and involves many more structures than either of the previous 2 senses. We're going to start with the accessory structures of the eye. Again, these are ones that help the eye function, keep the eye healthy, but do not really have anything to do with vision ability. You have 1, your eyelids. These are going to keep your eyes lubricated and free of dust. Attached to the eyelids are the eyelashes that keep out foreign matter, and also the eyelashes are associated with tarsal glands, which produce a modified sebum. Sebum is an oil. These keep the eyelids from sticking together or from sticking to the eye itself when you're opening and closing your eye. The third accessory structure is called the lacrimal caruncle. Lacrimal refers to tears. Your lacrimal caruncle is a mass of soft tissue composed of skin covering over some glands. It's over in the corner of your eye, the medial corner. The glands are sweat and sebaceous glands, and these glands produce thick secretions, and they leak overnight. And because they're leaking overnight and your eye is closed, you start to get an excess of these secretions, and they form gritty deposits in the corner of your eye. This is what you say when you say the Sandman came. It's really just secretions from the lacrimal caruncle. The fourth accessory organ is the conjunctiva. This is a mucous membrane that covers the epithelial tissue of the surface of the eye as well as underneath the eyelids, so that when your eye is closing, the conjunctive on the eyelid is touching the conjunctiva on the eye, and those 2 are rubbing together. They're going to keep the eyelid from sticking closed, provide some lubrication. If you've ever had pink eye, where it's an infection of the conjunctive, it's also called conjunctivitis. Your lacrimal apparatus is made up of a number of structures that are involved with producing, distributing, and removing the tears. Your tears are going to reduce the friction between your eye and your eyelid, remove dirt, dust, and bacteria, and also provide nutrients and oxygen to the conjunctival epithelium that's lining the surface of the eye. There are 4 structures involved in the lacrimal apparatus. The first one, the lacrimal gland, is going to produce the tears. This is going to be a watery solution that contains lysosomes and antibodies to kill microbes. You produce about 1 milliliter of tears per day, and the tears, as they flow across the eye, will mix with the tarsal secretions and make an oil slick on the surface of the eye to help with lubrication and slow down evaporation. Your lacrimal gland is located above the eye on the lateral side, and tears are going to move from the upper layer down across the eye to the medial lower side of the eye. Once it gets to the corner of the eye, the tears will enter into the second structure, that is the lacrimal canaliculi, which is going to be the canal where the tears drain out from in that lower medial corner. They will drain into the third structure, the lacrimal sac, and they will then move from the sac into the nasolacrimal duct, the fourth structure. The nasolacrimal duct is going to drain the tears from the lacrimal sac into the nasal cavities. So you produce about 1 milliliter of tears every day. When you cry, your emotions trigger the lacrimal gland to get overactive, producing too much fluid, and then it drains into the nasolacrimal duct, but it backs back up and comes back out the nasolacrimal duct and the lacrimal caniculi, and then rolls down the face. This is why you produce tears, and also why your nose runs when you cry. The eye itself is made up of 3 layers. These layers are in the eye wall, and they're called tunics. Your eye is a hollow ball with the 3 tissue layers forming the outside. The outermost layer is called the outer fibrous tunic, okay, and it contains 2 parts, the sclera and the cornea. The middle is the intermediate vascular tunic, and this is going to be broken down into the choroid, the iris, and the ciliary bodies. And the innermost layer is the inner neural tunic, which will be the retina, that has both neural and pigmented layers. In addition, we have 2 cavities in this hollow eye. The anterior cavity is going to be in the front portion of the eye and is going to contain a fluid called aqueous humor. And the posterior cavity is going to be behind, in the back of the eye, and will contain a fluid called vitreous humor. The fibrous tunic is the outermost layer, and its main function is for protection because it's a dense fibrous connective tissue, and it is an area where the extrinsic eye muscles can attach here so they can assist in focusing the light. There are 2 parts of the fibrous tunic. The major portion of it is the sclera. This will be coming from the back of the eye towards the front, and it is the white of the eye, made up of the dense fibrous connective tissue. It has small blood vessels in it, and its function is for protection. In the very front of the eye, the sclera gives rise to the cornea. The cornea is a clear, transparent portion of the eye. It has no blood vessels, so it depends on nutrient diffusion from the tears. Okay. It has got numerous nerve endings. It's the most sensitive part of the eye, and its function is to allow the light to enter into the eye. Corneal damage can lead to blindness, but corneal transplants are very possible since there are no blood vessels, therefore there's no white blood cells. The tissue is not rejected. The next layer, the middle layer, is the vascular tunic, also called the uvea. It has a number of functions. It is involved with secreting and absorbing the aqueous humor of the anterior cavity. It will supply the eye with nutrients. It will regulate the amount of light entering the eye, and it will adjust the shape of the lenses. There are different parts involved with these different functions. In the vascular tunic, we have the iris. The iris is in the front of the eye. It's a colored portion of your eye, if you have blue eyes or green eyes or black eyes. And it's actually a muscle. It's made up of 2 different muscles. There's the pupillary constrictor muscles that decrease the size of the pupil. That's the dark hole in the middle of the eye and the iris that we regulate. and pupillary dilator muscles that increase the size of the pupil. Now we're going to change the size of the pupil to regulate how much light enters into the eye. If it's dark, you're in a dark room, your pupil will get larger so you can get more light to come in. If you go into a bright room, your pupil will constrict. Okay. The iris also contains pigmented cells which are responsible for the color of the eye, plus it has blood vessels. The anterior surface of the iris is going to be made up of melanocytes, and the posterior surface is going to be covered with a pigmented epithelium. So your eye color is going to depend on the number of the melanocytes and the density of that pigmented epithelium. Few melanocytes and a thin pigmented epithelium, you will have blue eyes. As it gets thicker, your eye color will move from green to brown to black. The second structure in the vascular tunic is the ciliary body. These are little muscles that are going to be on the outskirts of the outer edge of the iris, and they attach to, they're right underneath the iris, they attach to the lens, which sits right behind the iris, with these little suspensory ligaments. And they're going to hold the lens in place behind the pupil and adjust the shape of the lens. So when the muscle contracts, it can alter the shape of the lens by pulling on the suspensory ligaments. The third layer is the choroid layer. Now this is going to be towards the back of the eye, and this is going to be right underneath the sclera. So it goes sclera, then the choroid. And the sclera in the front gives rise to the clear cornea. The choroid in the front gives rise to the iris, and then the pupil is in the center of that. The last layer is your neural tunic. Neural because it's involved with nervous tissue. This is the retina, the innermost layer of the eye. And this is going to be made up of two layers. It has a pigmented layer to absorb the excess light, which is located near the choroid, and then there's a thick neural layer, okay, on the innermost surface. Within the neural tunic, we have, or within the thick retina, we have photoreceptors. These are going to be closest to the pigmented layer. They're in the neural layer, but closest to the pigmented layer. You have two photoreceptors, your rods, which are for dim light and black and white vision, and your cones, which are color vision and bright light. We have three different types of cones. Also found in the back of the retina is an area called the macula lutea. And this is the area where the light would shine straight through the lens and go straight back and hit that spot in the back of the eye. That macula lutea looks sort of like a little dent. And in the very center of the macula lutea is the fovea centralis. Now your macula lutea and your fovea centralis are made just up of cones, but there are even more cones in the fovea centralis, and that's going to give us the best visual acuity, or best vision, that we can have. Also on the retina is the optic disc. This is where your optic nerve will exit the retina, and where it does, there are no photoreceptors, so this is considered your blind spot. So if the image focuses on that spot and stays on that spot, you would not detect it. However, when you are focusing on things, the image bounces back and forth around the eye continuously, so even if it hits the blind spot, it's there for so long that the other cells override it. Your anterior cavity is from the cornea to the lens, and it will be filled with aqueous humor, which is a watery structure, and it will possess nutrients and do waste transport. The posterior cavity, which is going to be from behind the lens back to the retina, is filled with a vitreous humor, a gel-like fluid, and this is mainly there to stabilize the eye shape, plus provide some physical support to keep the retina in place. And your lens, which is right behind the pupil, is going to focus the image of the incoming light on the photoreceptors. And it does this by being able to change its shape. So when it's looking at something up close or far away, the lens bends differently, so it can make sure all those light rays go and hit back right on the fovea centralis. And this movement of the lens is done by the suspensory ligaments. Your cells are made up of a number of cells of the lens are filled with crystalline proteins. They have no organelles or nucleus, so they give you a fairly crisp vision. In this figure, we're looking at the pupillary muscles, the constrictors and the dilators, and they are going to adjust the size of the pupil. Again, the pupil is regulating how much light comes in. So really the pupil doesn't do it, the iris is regulating it, because the iris is what's made of the muscles that open and close the pupil to allow light in and increase the amount of light if you're in a dark situation, decrease the amount of light if it's a bright situation. The retina in the back of the eye is going to be where the light is going to be detected, and it's going to be made up of a number of layers. Starting from the anterior of the retina, where the light will first enter as it moves towards the back of the eye, it hits the surface of the retina. That's the anterior of the retina. We have one layer of ganglionic cells. These are neurons whose axons will come together and form the optic nerve as it exits the retina. Then the second layer are your bipolar cells, again another type of neuron. And then the third layer will be your photoreceptors. So your photoreceptors are going to be the receptors that respond to the presence of light. They are going to then stimulate the bipolar cells, who will then send an action potential to the ganglionic cells that will then be taken to the brain. Okay, this is a little bit backwards, the way our structure is, because it would have been much easier to put the photoreceptors in the front, and then as soon as the light hit it, we already started processing it. So think of it as though I had to walk down the aisle of students, and I had to walk to the back of the aisle, tell them something, and then they had to turn around and pass that information back up to the front row, instead of me starting with the front row and sending the information straight back. So this is going to cause a little bit of delay in some of the stimulus. Okay. In this photo we can see the macula lutea with the fovea centralis in the center, which gives you the best visual acuity. And then you can also see the blind spot, the optic disc. This is where the axons of all the ganglionic cells are going to gather and form the optic nerve and then exit through the retina, back out through the choroid and through the sclera to leave the eye. And where that is happening is no photoreceptors at all, because the nerve is there, so we have a blind spot. In this figure we can see the organization of the retina that we just went over. Your neural layer is going to be the anterior surface, and it is going to be made up of the neurons and the photoreceptors, ganglionic to bipolar to photoreceptors. Right behind that neural layer of the retina is a pigmented layer of the retina. And this tissue is basically there to absorb any excess light so it doesn't bounce back into the eye, and then we get a real funky vision of different photos coming in at the same time. Behind your pigmented layer of the retina, we have the choroid, and then we have the sclera. And the optic nerve showing the axons of the ganglionic neurons forming the optic nerve and exiting through the whole retina and out of the eye, and producing the optic disc or blind spot. The anterior cavity is going to be filled with aqueous humor. Aqueous means watery, humor means fluid. And this is secreted by the cells of the ciliary body. The ciliary body is not just a muscle, but also has epithelial cells that secrete this fluid. It's going to flow through the pupil between the anterior and posterior chambers of the anterior cavity. So it's going to be in the front of the eye, behind the cornea, in front of the iris, then it will flow through the pupil and bathe the surface of the lens. It's going to provide nutrient and waste transport, as well as a fluid cushion for the front of the eye. It will be reabsorbed at a canal at the base of the iris and return to the veins in the sclera. We replace that old aqueous humor with new fresh aqueous humor on a regular basis. Your posterior cavity is going to be filled with vitreous humor, which is a gelatinous fluid. This is not replaced like the aqueous humor is, and its main function is to fill up that hollow ball with a gel-like material so it can stabilize the shape of the eye. Otherwise, light won't be bent properly, and we won't hit the fovea centralis. The lens is held in place by suspensory ligaments that extend from the ciliary body of the choroid. The lens is going to be able to focus the image on the photoreceptors of the whatever it is you're looking at. And it's going to do this by changing shape depending on whether the image is close to it or farther away from you. Okay. When light enters the eye, it's going to be bent or refracted at the cornea, surface of the eye, and again at the lens. This is going to hopefully bend the light so it goes toward the focal point on the retina, which is the specific spot of the intersection on the retina where the light will hit. The distance between the lens and the focal point is called your focal distance. And this is going to depend on the distance from the object to the lens. Your focal distance will increase as the object moves closer due to changes in the corneal shape. And it will also depend on the shape of the lens. The lens will round up to decrease this focal distance as something moves closer. Accommodation is the automatic adjustment of the eye to give us a clear vision. This is done by changing the shape of the lens. It will, and this is with the suspensory ligaments and the ciliary bodies pulling on it. The lens will round up to focus on objects that are close and flatten out to focus on objects in the distance. Now this focusing and changing of shape is going to bend the light wave images so that those light rays will come back and hit on the fovea centralis, hopefully, in the center of the retina so you have the best vision. Not always does that work, however. Accommodation, that automatic adjustment of the eye to give us clear vision by changing the shape of the lens so it rounds up to focus on objects closer and flattens out to focus on objects in the distance, is carried out by the ciliary bodies. These are muscles that make the lens round up. With age, those muscles and ligaments and the lens start to lose their elasticity and their ability to do this. So it doesn't round up as well, and the ability to see things up close decreases. We lose some of our natural nearsighted abilities. This aging process results in a condition called presbyopia, which is the natural decrease in nearsightedness. You become farsighted, and this occurs around age 40 when you start to need reading glasses. Normal vision is called emetropia, and this is when the ciliary muscles are relaxed and the distance image is focused on the retina surface, so you see things very clearly. Myopia is nearsightedness. This is when the image of a distant object is projected in front of the retina, so it appears very blurry. Nearsighted means you can see things close, but you can't see things very well far away. Hyperopia is farsightedness. You can't focus well on near objects. They look kind of blurry, but you do see things far away clearly. Another type of vision problem is astigmatism. This is a variation in the curvature of the lens or cornea. So instead of it being a nice round curve, it's kind of bumpy, and therefore it distorts the image and it focuses it back on the back of the retina in a blurry pattern. An image is going to be made up of many light points that project the image upside down or right and left reversal on the back of your retina. Your brain then will compensate for this and flip it back the right way so you have the correct orientation. Visual acuity is your clarity of vision. A person who has 20/20 vision has normal vision. They can see certain details at 20 feet from the object, and that's considered normal. If you have 20/15, you can, at 20 feet, you see better and with more clarity than the normal vision did. And if you have 2200, you see much worse. What you can see at 20 feet away is what a person standing 200 feet away from the object with normal vision can see. If your vision is worse than 2200, you are legally blind. Visual physiology. We're going to detect different wavelengths of different colors with our photoreceptors, our rods and our cones. The rods detect packets of light called photons, but they can't detect different colors, so they're going to see in black and white and very dim light. Your cones can distinguish between different wavelengths, thus they detect colors. There are red, blue, and green cones, and they detect bright light. Your photoreceptors, both your rods and the cones, are made up of two parts. There's an outer and an inner segment. The outer segment is going to contain discs that have little flattened discs that contain rhodopsin, which is a visual pigment. This is going to be made up of opsin, a protein, and a molecule of retinal, which is a pigment made from vitamin A. There are different types of opsins in the 4 different photoreceptors, the 1 rod and the 3 different types of cones, and they detect different wavelengths. These discs are going to be constantly replaced from the bottom. The new ones are being made, and they move up the stack, and as they move closer to the surface and get to the top of the stack, these older discs are going to be released through exocytosis and be gotten rid of. In the darkness, your photoreceptors are continuously stimulated, and they're stimulated by an inhibitory neurotransmitter. And so that in the darkness, they're inhibited. So when there is no darkness and there's light, we stop the transmission of the neurotransmitters. They're no longer released, which allows the photoreceptors to see. Okay. With photoreception, the photon is going to hit the receptor. And when it hits the receptor, it's going to have the retinal, which absorbs it, activate the opsin. This is going to stop the releasing of the inhibitory neurotransmitters. This is now going to allow the bipolar neurons to be stimulated. After stimulation, the rhodopsin is going to be broken down and recycled again. Color vision, we have cones, red and blue and green, and they detect the different wavelengths. And they are going to integrate all this incoming information to come up with your full color vision. Color blindness occurs when one or more of those cones are non-functional. Most common type of color blindness is red-green color blindness. Tends to occur more in males. There is also a color blindness that affects the blue cones. Your visual pathway. This is how the message is going to move. Once the message starts, it's got to cross those two synapses. The photoreceptor picks it up and is excited. Then it passes it to the bipolar cell, who then has to pass it to the ganglionic cell, and then it will head to the brain. This is going to increase your synaptic delay and provides more opportunity for processing and integrating visual information before it leaves the retina. Now, your vision is going to vary. Your rods see blurry vision, and this is because there's a much convergence occurring in the rods. You have 130 million rods that then pass their information to 6 million bipolar cells, and that passes to 1 million ganglionic cells. And this results in a very blurry vision. With your cones, however, there is no convergence. So it basically goes from the cone right to one bipolar, right to one ganglionic cell. So we have a very crisp and colorful image. The final of the special sense organs is your ear, and this is going to be used for both hearing and for equilibrium. These receptors are going to be simple mechanoreceptors, and they are going to be made from hair cells that have little hairs or cilia on them, and they are going to actually bend when they bend, which is a mechanical change, that will stimulate the neurons to then start the action potential. Your ear is going to be broken down into three parts: your external, middle, and inner ear. Your external ear is going to be air-filled, and it's going to start with the outside of your ear, your cartilaginous pinna or auricle. This is where you pierce it. And this job is going to be to funnel the sound into the external acoustic canal, or meatus, either term. Okay, it's going to funnel the sound into it, then it's going to head down that auditory canal until it hits the tympanic membrane, which is your eardrum. This is going to be a sound wave that goes down, and then the wave will be passed from the eardrum further into the ear. The canal is going to contain ceruminous glands, which are special sebaceous glands that secrete cerumen, which you call earwax. And the function of the earwax is to trap foreign particles, and also it can inhibit the growth of microbes. Your ear canal or auditory canal will also contain hairs that will help trap foreign objects from getting deep within the canal. Your middle ear is also going to be an air-filled chamber, and it is going to extend from behind the tympanic membrane to the oval window, which is a membrane, another membrane that's on the cochlea, which is the inner ear. Okay. This is going to be an air-filled space. It's going to have a connection from this space down to the back of your throat via the auditory tube, and that is going to work as a pressure equalizer. Okay, when you go up in pressure or go down in pressure, say you go up in an airplane, the pressure outside the ear in the external auditory tube versus in the middle ear, that air pressure changes. Originally when you started they were both the same. Now there's a change in pressure, and that change in pressure is going to cause pain. And if nothing can happen to correct that, your eardrum, your tympanic membrane, will actually rupture to equalize that pressure. Instead, you have the auditory tube which goes down to the throat, and you'll yawn or you chew gum, and that will pop it open, and that will let the air come in through the mouth, come up there, and now you have the equal pressure of air on either side of the tympanic membrane. Okay. You also have within that middle ear 3 auditory ossicles, ear bones, the malleus, the incus, and the stapes. And these are going to connect the tympanic membrane to the oval window. You're going to pass that vibration from the tympanic membrane to the malleus, which then touches the incus to pass the vibration, which touches the stapes, who passes the vibration to the oval window. Along the way, it's going to not only pass the vibration, but also amplify the sound. And the third part is your inner ear. Now, this is a network of fluid-filled tubes and chambers. This is going to include your semicircular canals, your vestibule, and your cochlea. The first two, semicircular canals and vestibules, are going to be involved with equilibrium. Your cochlea is going to be involved with hearing. Your vestibule is going to be broken down into 2 sections called the saccula and utricle, and these are involved for detection of gravity and linear movement. The semicircular canals are going to be involved with detecting angular movement, and your cochlea, which is a spiral-shaped, sort of like a snail shell structure, is going to contain the receptors for hearing. These will be your hair cells with little tiny hairs called stereocilia. Think of stereo for your hearing. Okay. The sound vibrations are going to be transmitted from the stapes into the oval window to the fluid inside that cochlea, and now the fluid will start a wave, and that vibration will be passed up the cochlea and back down to the round window, which is the last membrane that just acts to dampen the pressure. Along the way, that fluid will bend the stereocilia of the hair cells, and that will then detect that pressure. And those are the mechanoreceptors. Equilibrium is going to be detected by the semicircular canals and the vestibules. Okay. The semicircular canals have little hair cells inside the duct that respond to the rotational movement of the head. There are 3 different semicircular canals, and each one is sensitive to a different plane of motion. So if you move your head to say yes, or to say no, or to tilt your head from side to side, one of those 3 different semicircular canals will detect this motion. Within the semicircular canals, there is an enlarged region that's called the ampulla, okay? And this is where your receptors are going to be found. In that ampulla, there is going to be a gelatinous structure that is called the ampullary cupula, and in that gelatinous structure we have that's going to sit right on top of your hair cells and the stereocilia. So your stereocilia sit in this gel-like structure, and then surrounding the gel-like structure is fluid. And when you move your head, that fluid starts to move, causing a pressure wave which pushes on the gel, pushing it a little bit, and when the gel moves, that then bends the hairs, the stereocilia, and you detect motion. Again, that endolymph fluid of the semicircular canals is going to move. It's going to push on that gelatinous ampullary cupula, which will then bend, and when it bends and moves, it will bend the stereocilia, and those stereocilia bending stimulate the hair cells, which are the receptors, and you detect angular motion. The vestibule is made up of the saccule and the utricle. These receptors are going to provide sensation for gravity and linear acceleration and deceleration. The saccule is going to respond to vertical movement, where the utricle will respond to horizontal movement. With this structure, it's the same concept of the hair cells embedded in a gelatinous structure and then bending over. In this setting, the hair cells are going to be, in their processes, are going to be embedded in a gelatinous membrane, and the membrane is called the otolithic membrane. And sitting on top of the membrane are little otoliths. These are calcium carbonate crystals, which provide a little weight to the membrane. And they sit on the membrane, and when you move, those otoliths will shift a little bit. And they'll move back and forth. If you run and stop quickly, they'll slide back and forth. And the otolith shifting is what's going to bend the hair cell processes and trigger the reception. Hearing is going to be detected in the cochlea of the inner ear. It has hair cells with little stereocilia on it. Now your sound is going to move through air or water as a wave of pressure. Frequency is a measure of waves per second. We measure that in hertz. And we perceive this frequency as a pitch. A high-pitched sound is short waves, and a low-pitched sound is more of long waves. Your energy content of the wave determines its intensity or volume, measured in decibels. Sounds greater than 120 decibels can break the stereocilia off the hair cells and cause hearing loss. This is why if you listen to really loud music, that could cause deafness in later years. Your cochlea is a spiral-shaped structure. It looks sort of like a snail shell, with the bottom being bigger than a smaller layer, than a smaller circular layer. Okay? It's going to be made up of 3 different ducts or tubes that are going to spiral up the cochlea. So they're going to start in the bottom layer and then move up to the second layer, then move up to the third layer. Okay? The 3 ducts are called the scala tympani, which is the bottom duct. This is going to be filled with perilymph, which is similar to cerebral spinal fluid. The scala vestibuli, which is the top duct, filled with perilymph. And then in between those 2 layers, there's a much smaller duct sort of squished to the side, and this is called the cochlear duct. And this is where your sensory receptors are going to be located for sound and for hearing. This is filled with a different type of fluid called endolymph. Now, if you think of it this way, the scala, think of it sort of like a parking garage. You go up the up ramp, you keep going up to the top, and then you come down the down ramp. This is the same way the cochlea works. The scala tympania goes up to the top of the cochlea, so the sound wave will travel up the scala tympania, and then it will turn the corner at the top, and it will travel back down the scala vestibula. Okay? And it will bring it back down to the bottom of the cochlea. At the beginning of the scala tympani is the oval window. At the end of the scala vestibuli is the round window, which works as a pressure dampening device. Located in the cochlear duct is a structure called the spiral organ, or the organ of Corti. And in this little structure is where we are going to actually find the receptors for hearing and detect the sense of hearing. The spiral organ, or organ of Corti, is made up of 3 things. You have a bottom membrane called the basilar or basal membrane. Bottom and basal both start with B. And this is going to be found right above the scala tympani, the bottom duct layer. Then we have on top a tectoral membrane. Tectoral means to touch. Tectoral and top both start with T, so it hangs out over the top. So the basilar is the bottom, the tectoral is the top. On the basilar membrane, we have the hair cells with their little stereocilia sticking out. Those are going to reach up towards the tectoral membrane. The hearing process is going to be broken down into steps. The first step, the sound wave moves into the external acoustic canal and hits the tympanic membrane. Second, the tympanic membrane will vibrate, and that will vibrate with sound waves between 20 to 20,000 hertz, and will pass this vibration to your auditory ossicles. First the malleus, which is touching the tympanic membrane, and the malleus also touches the incus. It will pass it to the incus, which will then pass it to the stapes. Along the way, it not only transmits the vibration, but amplifies the sound as it goes in. The third step, the stapes will pass that vibration to the oval window, the entrance to the cochlea. This is going to go into the oval window, and that pressure is going to go into the perilymph, the fluid of the scala vestibuli, which is the top chamber, the top ductwork. It's going to travel up the scala vestibuli and then back down the scala tympani. Okay. Now, as it's moving down the scala tympani, there is, right above the scala tympani, the basilar membrane of the cochlear duct, which is wedged between those 2 vestibuli and the tympani scalas. Okay. So as it's traveling underneath the basilar duct, the pressure wave is going to push, underneath the cochlear duct, it's going to push on that basilar membrane and push it upward. This is going to cause the stereocilia of the hair cells that sit on the basilar membrane to rise up and come in contact with the tectoral membrane. They touch it, and this will actually cause those stereocilia to bend. When the stereocilia bend, they open ion channels. and opening of the ion channels causes depolarization. The number of cells depolarizing at one time will determine the volume of the sound, and the pitch is going to be determined by where that pressure wave crosses from the top to the bottom chamber, the scala vestibuli to the scala tympani. I said before it went all the way to the top. That is only the case it's a very low frequency sound. If it is a higher frequency sound, it will cross over earlier, and therefore stimulating different parts of the basilar membrane and different parts of the cochlear duct. Information about frequency and intensity is going to be relayed to the auditory center of the central nervous system.